Microbial fermentation is used in the production of many different foods and beverages, including yogurts and cheeses, soybeans, chocolate, and beer and wine. In the absence of oxygen, fermentation completes the catabolism of glucose. Glucose catabolism begins with glycolysis, an ATP-generating pathway that produces pyruvate as an end product. To complete catabolism, glycolysis requires NAD+ to accept electrons that are removed from the sugars. This means that the cell needs to continually regenerate NAD+, or glycolysis will grind to a halt. To regenerate NAD+, in the absence of oxygen, NADH from glycolysis must transfer electrons back to pyruvate. In alcoholic fermentation, enzymes first remove carbon dioxide from pyruvate, forming acetaldehyde, then transfer electrons from NADH onto acetaldehyde, forming ethanol. In this way, NAD+ is regenerated. The baker's yeast, Saccharomyces cerevisiae, performs alcoholic fermentation. The yeast cells excrete the alcohol into their environment, such as inside a vat in a brewery. The excretion of a product that still has the potential to yield energy for the cell may seem wasteful, but without oxygen or other molecules to accept electrons from ethanol, the cell has no choice. These same cells conduct small amounts of other reactions besides the main fermentation pathway. These other reactions provide the alcoholic product with flavors. Some chemicals from these other reactions produce off flavors, while others produce desirable flavors. Different species perform different types of fermentation. Some, such as Lactococcus lactis, perform lactic acid fermentation, regenerating NAD+ by creating lactic acid. Lactic acid fermentation converts milk into yogurt and cheese. Humans also perform lactic acid fermentation. Muscle cells produce lactic acid during exertion when oxygen becomes depleted. In foods, fermentation preserves food value because the alcohol or acids produced by the microbes also limit continued microbial growth, while leaving most of the protein intact.